This is ISCI 2001, Chapter 20, Part C, The Nature of Living Things. There are 4 themes for today's lecture: respiration and photosynthesis. Then there's the importance of cell division. Then we'll talk about the cell cycle. And then last will be the stages of mitosis. All right, so we will start with respiration and photosynthesis. All cells use a molecule called ATP. This is important to know. We call it adenosine triphosphate, and it is the energy currency to carry out every function in every cell. Okay, ATP, it is important to notice or to note that ATP is the usable energy in all cells. Okay, the usable energy. So ATP gets the job done on a cellular basis. This picture that we have here at the bottom, this is one ATP. Okay, you have adenine here, you have ribose here, and then you have the phosphate groups. How energy is released is, if you look closely, you have 3 phosphate groups here. That's where we get the word triphosphate, because tri means 3. Okay, tri means 3. What happens is one of these bonds is broken. Energy is released when a bond is broken. Okay, so this adenine with the ribose and the phosphate groups, adenosine triphosphate is converted to adenosine diphosphate. So the 3 becomes a 2 whenever this bond is broken. So then you have adenosine diphosphate, and it's kind of like a rechargeable battery, because whenever you have a rechargeable battery, you can plug it back in, the battery will recharge, and what is going to happen is this bond will be reformed. They'll find a new phosphate to bond with. We'll once again have our 3 phosphate groups, and it will become adenosine triphosphate once again, and it will be a brand new battery with the usable cellular energy that we can use. How do cells make ATP in order to carry out necessary function? Through 2 processes. We have cellular respiration, and then we have photosynthesis. Okay, if you don't have a chloroplast, you are not going to carry out photosynthesis. However, we are animals, we consume our food, therefore we do carry out cellular respiration. So if you have chloroplast and you carry out photosynthesis, such as green algae or plants, then you will have to do both photosynthesis and cellular respiration. But since we eat our food and we don't make our food, we only have to do the cellular respiration. Let's talk about the cellular respiration. It is a series of extremely chemically complex reactions that happen within the cells to produce ATP. It happens in 2 places. It's going to happen, first it will happen in the cytoplasm. That's where it starts, in the cytoplasm of the cell. And then it will move, or we want it to move, into the mitochondria. Oh, I'm running out of space. Let's erase this. I'll write it a little bit prettier, okay? So it starts in the cytoplasm. That's where the anaerobic respiration happens. And then if you are breathing, which we hope you are, it will then move into the mitochondrion. There we go. That's better. So the starting molecule is going to be glucose, which is what we eat, or if you're a plant and have chloroplasts, it is going to be the sugar molecule that is created whenever you convert that solar energy into chemical energy. If oxygen is available, then aerobic respiration is going to happen, and you're going to create 36 pieces of ATP, so 36 molecules of ATP for every 1 piece of glucose. That's a lot. If no oxygen is available, then you're only going to get 2 molecules of ATP from anaerobic respiration. So the 2 pieces from anaerobic plus the 36 pieces from aerobic respiration, a total of 38 pieces of ATP altogether, if you're breathing. We want to be breathing. And you may be saying, well, plants don't really breathe, but they do have the things necessary that they need for aerobic respiration. They do have oxygen around them, so don't worry about them. They've got what they need. They are not in an oxygen-deprived environment. So here what we have is this first picture here is going to be glycolysis. Okay? With glycolysis. Okay? Glycolysis the word can be broken down into two things. Glyco is glucose. Okay, that stands for glucose. Lysis means to break down. So the whole point behind glycolysis is to break down this glucose. Okay? So it's going to be just this whole series of steps, basically to break down glucose into pyruvate, or another word that you can call it is pyruvic acid. Okay, so lots of complex steps. You're not going to have to teach elementary kids all of these steps. Basically, what they're going to have to know is it's a series of steps to get from here to here. Okay, is what glycolysis is. It's our bodies breaking it down. And this is what happens in the cytoplasm. It does not need oxygen to happen. Okay? The rest of it does. So, once we get to the Krebs cycle, okay, this is where we're going to need the oxygen. Okay? And let me back up for a second. It's important to note that the chemical compound, the chemical equation for glucose is C6H12O6. Carbon, okay? Anything that contains carbon, we call it organic. This is not the grocery store organic or the organic that you hear in the media. This is organic as far as science is concerned. Anything containing carbon is organic. So when I'm talking about organic carbon, this is molecular organic. If you were to take organic chemistry, okay, if you've heard of that course, this is chemistry that contains carbon. So anything living has carbon in it. You have carbon, I have carbon, anything living has carbon. So carbon is very important to life. Okay, so once we've gotten this C6H12O6, this carbon or this glucose broken down into pyruvate or pyruvic acid, all this citric acid or Krebs cycle right here is showing is, we call it the citric acid cycle, is because if you see this word here, citric acid, it's got 6 carbons in it that I just completely erased. It's all being broken down. It's going to form NAD plus. This basically what's happened is there's going to be 6 NADHs. They're generated per 3 per acetyl coenzyme A that enters. There's 2 FADH2s that are generated per 1 acetyl coenzyme A that enters. There's 2 ATPs that are generated per 1 acetyl coenzyme A that enters. There's 4 CO2s that are released. I mean, it's a whole big messy cycle that is very intense. The main bulk of everything here that generates the most energy is going to be this right here. And this is the electron transport chain. The whole goal of this electron transport chain is to break down all of that NADH and the FADH2, pumping hydrogen ions into the outer compartment of the mitochondria. It happens in the mitochondria. So does this Krebs cycle or citric acid cycle. In this reaction, the electron transport chain creates a gradient which is used to produce this ATP, kind of like the chloroplast. It typically is going to produce 32 of the ATPs. The citric acid cycle is going to produce 4 of the ATPs. The glycolysis is going to produce 2 of the ATPs total, so that's where you get all of your 38 total. So this is how your body produces all of those ATPs. It's a huge, huge, huge complex thing, but long story short, if your body does, doesn't receive the needed oxygen and only goes through the first stage, which is anaerobic respiration, the glycolysis, you'll undergo cellular death, and because you'll only be making 2 pieces of ATP instead of the 38 pieces of ATP, therefore your body will go through that ATP pretty quickly, and it will result in brain damage and then death. Okay, what we have in this slide is just a picture of anaerobic respiration. What we have is glucose is needing to be broken down into the pyruvate. It's going to take 2 cycles here to make the pyruvate, and basically what needs to be happening is the goal is to reduce the pyruvate, thus generating the NADP+. In a single cellular organism, what happens is alcoholic fermentation. It happens in the cytoplasm. This occurs in yeast and other types of bacteria. The product of fermentation is alcohol, which is going to be toxic to the organism. And the alcohol is ethyl alcohol. This is used in making things like yeast bread, beer, wine, other alcoholic beverages that we use. And then in multicellular organisms such as animals, so humans and such, it's going to be the lactic acid fermentation. The product of lactic acid fermentation is lactic acid, which is going to be toxic to mammals. This is the burn felt when undergoing strenuous activity when you work out. So this is the burn you feel when you're working out. And this also contributes to the sore feeling that you get the next couple of days after you have worked out. Let me back up real quick and just say that the only goal of fermentation reactions is to convert the NADH into NAD plus to use in glycolysis. All of this happens only in the cytoplasm and not in the mitochondria, and no energy is going to be gained from this. Note the differences. Anaerobic respiration is 2 ATPs produced from glycolysis. Aerobic respiration is 36 ATPs produced from glycolysis, Krebs cycle, and the oxidative phosphorylation. So the evolution of an oxygen-rich atmosphere, which facilitated the evolution of aerobic respiration, which was crucial in the diversification of life. This is the chemical reaction for aerobic cellular respiration. We start out with 1 piece of glucose, 6 pieces or molecules of oxygen. It's going to yield 6 molecules of carbon dioxide, 6 molecules of water, and of course energy. So, and there's it written out, and this is the balanced equation. I want you to note that in both sides we do have our organic compound carbon. That will be important, and I want you to also note the compounds: glucose, oxygen on one side, carbon dioxide, water on the other side. It'll be important when we talk about photosynthesis next. So photosynthesis, this is the process that only occurs in plants and in some algae. It takes place in the chloroplast of plant cells, whereas cellular respiration occurs in the mitochondria. This is the original source of energy, comes from sunlight for chemical reactions driven by photosynthesis. Remember, we eat our food, animals eat our food, plants and other algae have to use the sunlight to obtain the solar energy to be able to convert that energy into chemical food. Very simplified version that you can give to your students is water plus light equals chemical energy. So what happens is, first the chloroplasts are going to trap the light energy. Water is going to enter the leaf from up in the roots that flows up through the xylem part of the plant. Third part, carbon dioxide, enters through the stomata in the leaf, which is an opening on the underneath side. And then the fourth part is sugar leaves the leaf through the phloem part, transport part of the leaf. And so then you've got the chemical energy plus carbon dioxide equals sugar. So that's a very simplified version. Once again, this is the picture that we used last week in our lecture about the organelles of the plant. You've got your outer membrane and inner membrane of your chloroplast. You've got your granum, which are your stacks of individual thylakoids, and then your inner membrane space that is in between your inner membrane and outer membrane, and then your stroma, which is the space in between your stacks. Okay, remember I told you to remember the equation for cellular respiration. If you notice, once again, we have our glucose and oxygen, our carbon dioxide and water. This is the same exact equation, just flipped around. For photosynthesis, if you have light energy that's in the presence of a chloroplast with chlorophyll, once you add your carbon dioxide and your water, then it's going to yield glucose and oxygen. And once again, this is the balanced equation. So photosynthesis and cellular respiration, they form a continuous cycle. Part 2, the importance of cell division. Making new cells replaces dead cells. Dead cells with new cells. It repairs damaged tissues and allows living organisms to grow and develop. The functions of cell division is to divide, maintain the body for upkeep, and to repair for fixing things. Part 3 is the cell cycle. All eukaryotic cells undergo the cell cycle, which is essentially 2 major stages. First, you have interphase, which is for growth, and then you have mitosis and cytokinesis, which is cell division. Now, if we look at this as a cycle like this, as a big circle, your interphase is going to be basically all of it except for this little section here. So all of this in here is going to be your interphase, and this little slice right here is going to be your mitosis and cytokinesis. Okay, we're going to start here on this section here. Okay, interphase is going to be G1, which stands for growth and normal metabolic roles. It's going to be your first growth phase. After it goes through the first growth phase, then it has to replicate the DNA to go ahead and start preparing for nuclear division. And we call that the synthesis phase, because synthesis means to make. Once it has made enough DNA for DNA replication, it goes through the secondary growth phase for the preparation for mitosis, and we call that G2. So we have G1, S, and G2. After that, it is ready for mitosis. There are 4 different phases for mitosis, and you need to memorize these in order. It is prophase, metaphase, anaphase, and telophase. And at the end of telophase, that is where cytokinesis occurs. And cytokinesis is just basically where the cell divides apart. Interphase is divided into 3 separate periods of time. You've got G1, which is where the cell grows and gets larger. S is for the synthesis to make the DNA. It's where the cell replicates all the genetic information. And then G2 is where the cell grows a little bit more. All of this interphase lasts approximately 90% of the cell cycle. So you need to remember also that this is not talking about necessarily cell division, but nuclear division, especially when we're talking about the synthesis. We are talking about the synthesis of DNA. This is for nuclear division, so the division of the nucleus. This is what mitosis is for. Mitosis is nuclear division, division of the nucleus, and the cell divides along with that, of course, but the important part is that the nucleus correctly replicates all of the DNA that's inside of it so that cellular life can go on correctly, and that all of the DNA is correctly copied and replicated, divided, and then all of the cells can go on and correctly perform their cellular functions. Mitosis is the actual cell division where genetic information is halved. So, okay, here we have our genetic information is halved. That's our nuclear division, and forms 2 genetically identical daughter cells to that of the parent cell. The 4 steps are prophase, metaphase, anaphase, and then telophase with the cytokinesis. Prophase is where the nucleus breaks down and the chromosomes start to form. So here you have your cell membrane, your nucleus, or your nuclear membrane starts to break down. Your chromatin begin to start condensing so that you can see your chromosomes forming. Metaphase Your nuclear membrane is completely broken down. Your chromosomes are now at the middle. They're actually even paired together, and your chromosomes are in the middle, or the equator or the equatorial line of your cell. Then you have anaphase. The DNA replication is completed, and the chromosomes are going to be moving towards the poles of the cell. So they're going to be being pulled towards the poles of the cell. And then lastly, you have telophase and cytokinesis. You have two daughter cells that form along the cleavage furrow, and cytokinesis occurs when the two cells completely separate, resulting in two genetically identical daughter cells. So you have this here is your cleavage furrow. That's also the pinching in is where the cytokinesis is occurring. Your nuclear envelope is reforming. Your chromosomes are actually going back into chromatin, and your cell membrane is going to be closing back in to form your two daughter cells. So here are some nice pictures of that. Here's an artist depiction that is not O'Mara style. You've got your parent cell with the prophase going into metaphase. You can see them lined up here in the middle. Anaphase, they're being pulled to the side. Telophase. An easy way to remember all of these, prophase is just basically where the nuclear membrane is breaking down and there's one cell that's kind of stretched out and elongated. Metaphase, meta means middle, so that can be a word that can kind of help you remember that they're lined up in the middle. Anaphase, if you look at your chromosomes here, if you were to take this chromosome and it's turned like this, but if you were to flip it and turn it like this and then draw a line here, it looks like an A, okay? A stands for Anna. And then finally, telophase, it kind of looks like a weird-looking flip phone or an old-looking telephone that has the earpiece and the receiver here. But you can see that it's forming two daughter cells that you have there. You can see the cleavage furrow here, and that's also where the cytokinesis is occurring. Here are the real pictures that you're going to be seeing in the lab on Tuesday. Starts with prophase. You can actually start to see the chromosomes that are appearing, that are condensing, that's here in the nucleus. Metaphase, they're lined up. You've got your spindle fibers that are starting to pull them to either side of the cell. Anaphase, they are being pulled, and they look like two blobs that are being pulled to each side of the cell. And then finally, telophase, the way that you can tell that these are not two individual cells yet is that they do not have that nice nucleus that are surrounding the chromosomes. Okay, so those are not there, but you do have the cell membrane that is starting to form back. So the GPS standards that we covered today basically focused on energy flow. We were able to demonstrate the process and significance of photosynthesis, demonstrate an understanding of the need for oxygen and organic carbon, and explain the process and significance of cellular respiration. All right, so please do not forget. What am I going to tell you not to forget? Don't forget to go to the discussion board. Okay, don't forget to post your 3 discussion topics and or questions. Don't forget to post some meaningful conversation there. Y'all been doing a great job of that. Also, don't forget to go and take quiz number 3 before I post our next lecture next Thursday. So you need to do that before Wednesday night at midnight. Have that done. And I look forward to seeing y'all Tuesday at our lab. Make sure that you have read through that lab and you are prepared to look at all of the slides that we have prepared for mitosis, and I will see you then. Y'all have a great week.
Made with Doodlecast Pro.